Welcome everyone to the second rules session of our Regeneron STS webinar series. I'm Allie Steeple, the director of Regeneron STS, which is a program of Society for Science. I'm joined here today by my colleague Elaine Edwards, who can introduce herself.
Hi, everyone. My name is Elaine Edwards, and I am the Senior Specialist for the Regeneron Science Talent Search. Allie and I are so excited to have you here at our second of our rules mini sessions, today focused all on vertebrate animal rules.
That's right. And before we get started, we want to take a step back and welcome anyone who is here today because you're thinking of entering the Science Talent Search, you're mentoring a student, whatever it is. Great job to come here to learn a little bit more about our rules. And for you, we wanted to just give you that reminder that this year's application is now open for the 12th grade student living in the United States or U.S. citizens who are living abroad. The application is open until November 6th. So you still have several months to get in there and get going. But a little bit about STS, note that we cover a lot of this content at other sessions that you can find on our YouTube page if you're curious more about the program itself, the application, but just the general overview for those who are new to STS. STS is the nation's oldest and most prestigious science research competition for high school seniors. It's unique because we are looking for future leaders in STEM. We are not looking just for the best research, which is a very important aspect of our process too, but we're looking at kind of the whole student and what comes along with their research package. The students first come to us through an online application. So that's what's open now. You'll find the link at the end of this webinar session today. The application is free. It's essentially a college application with a 20-page research paper lumped on top and some essays about that as well. So you're coming to us if you're an entrant or an interested student with a research project already. You're going to want to make sure that it adheres to our official rules. So we'll let Elaine talk a little bit about that next.
Yes, so as Allie mentioned, we talk a lot about eligibility and process at all of our other sessions, which are available on our YouTube page. But this here is the second of our rules mini sessions, and these are really designed to kind of be a deep dive into three aspects of our scientific rules. Last week, we did a session all about human participant research. Today, we'll talk a little bit about vertebrate animals, and next week, we will talk a little bit about potentially hazardous biological agents and hazardous materials. But the reason that we want to do this is every year a number of entries fail to qualify because they do not meet the rules of entry. And so we really want to make sure that entrants and mentors avoid pitfalls after working so hard on a project. And we want to make sure that all humans and animals, the environment, all are kept safe during the student research process. We especially today want to provide clarity around our animal rules because these are different in many ways to the rules of our sister competition, Regeneron ISEF, which many people watching today might be a little more familiar with than they are with STS. It's also important to note when we're talking about these rules that while there are some state and federal laws that help keep some research consistent, every lab and every research competition does have their own sets of rules and regulations. And our STS rules, if you're submitting your project to us, need to be followed, even in cases where our rules might be stricter than your lab or what your mentor wants you to follow. So if you plan on entering STS, we do recommend that entrants, their mentors, and educators review our rules book annually. We make small edits to this every year, but all of the information that you will need in order to conduct your project successfully and in adherence to our guidelines is in that book.
That's right. And just to reiterate something Elaine said, we're going to reiterate this a lot in today's session, especially when it comes to working with live vertebrate animals. The STS rules are different than the ISEF rules. So if you are a younger student or a teacher who works with younger students and you're planning out your research project and you want to be able to use a project for both competitions down the line, you're totally allowed to do that, by the way. Please keep this in mind. This is one of the more common reasons that projects fail to qualify for STS. We love to see ISEF alumni and local science fair alumni applying to STS, always great candidates. But sometimes just that permissions piece or where you did the research makes a big difference in if you can actually apply. So we're going to get to that, but want to flag it now so you don't turn off your screen if that might apply to you. Great. So like Elaine said, we're talking vertebrate animals today, live animals and animal tissue or cells. We want to talk through what paperwork you might need, what pre-approvals that you might need to obtain like before you get started on the project. And we'll go through some examples. This should help you hopefully place the context of your project into our rules and understand better what you need to do. But just like in our past sessions, our examples might not be a perfect match to your project, and we encourage you to leave questions in the comments on our YouTube page, and we'll get back to them by next week and kind of leave a transparent trail so that students have answers to questions, and also so that, if you have a similar situation to another viewer, then maybe we'll answer that question for you. If you have questions today that you don't want to send in the chat or at any point in our process, we accept a lot of emails. We answer lots of emails every day from entrants to this program. You can always email our team at sts@societyforscience.org. We'll link that at the end of this session as well. So know that you're always welcome to email us if there's any question about the work you're trying to do. Also, before we get started, know that next week we're going to talk about PHBAs, potentially hazardous biological substances, and the rules that go along with those. A lot of vertebrate animal cell and tissue research and human cell and tissue research that's going to count or could count as PHBA research. So you might want to tune into that one too, just to make sure that you're being safe if you're working with bacteria or anything like that. So let's get started. We're talking animals today.
So the first question you're probably asking, what is a vertebrate animal? In general, the way that we define this is bones or no bones. But for the purposes of STS, a couple of things that we specifically define as vertebrate animals. So this refers to all non-human vertebrates. That includes fish at hatching or birth. It includes live non-human vertebrate mammalian embryos or fetuses, tadpoles, bird and reptile eggs starting 3 days or 72 hours prior to hatching, zebrafish past 7 days or 168 hours post-fertilization, and cephalopods, which are to be treated as vertebrate animals. So that refers to squid and octopi. If you're working with tissues from these types of animals, that is a tissue study, and we're going to talk about that a little later. But for now, we want to consider what you need to do if you want to study vertebrate animals while they're alive.
That's right. And one note about this slide, if you've had students enter STS before, cephalopods, treating them as inverts in our rules anyway, that's new this year. So to the point of our rules change every year, that is a new one. We want to be respectful of our very alert friends out there in the deep seas. All right. So one of the first things to think about if you're working with live vertebrate animals for research is where to do this work. This is where the rules start to differ between ISEF and STS. If you are working with live vertebrate animals or your project involves live vertebrate animals, your research must happen at an RRI, a regulated research institution. And to do work at an RRI, you have to find a mentor who works at one of these locations. The work probably needs to be either part of your mentor study, or the work that you're trying to do needs to be pre-approved before you get started. That work is going to be pre-approved by an IACUC, an Institutional Animal Care and Use Committee, which will be located at the institute. So what counts as a regulated research institute? You can Google this, but usually like your local university, local hospitals, any kind of federal lab, your high school is not an RRI. Okay, so you cannot be doing live vertebrate animal research at your high school. We're going to talk more about this as we go through, but know that if you're working with live vertebrate animals, it must happen at an RRI. There are a lot of rules about what you can do at that RRI. We'll talk about that next. The other place where you might do some vertebrate animal research is in the wild. What do we mean by that? We're going to talk about that separately too, but in very limited cases, if you are only observing, then you're allowed to do that work. And so we'll talk about what does it mean to only observe. It means you don't introduce any other stimuli to the environment that could change the animal's behavior. Or there's some citizen science that we do allow, and some things that you can do if you're performing citizen science. So we'll talk about that, but really those are the only two locations. So at an RRI or in the wild, not at your home, not at your school. There are some arguments to include those two things, but again, we'll talk about that next. So let's say you're at an RRI, you're lucky enough to find a position, which we know can be a limiting factor for STS. Also, just to backtrack, for ISEF, you have a little more freedom about where you can perform animal research and what you can do to animals, and you have to have veterinary supervision and be under the care of a veterinarian. That's not the case for STS. You really do have to do it at an RRI. So again, say you're lucky enough to be there. The projects that are involving experimental procedures that the student designed and conducted on vertebrate animals must adhere to all of the following things that appear on this slide. We're going to go through them over all together, and then we're going to deep dive into each one. First, so remember, all of these things must apply to the research that you're doing on a live vertebrate animal at an RRI. Any physical contact with the animal is restricted just to basic handling and husbandry. So what that could mean is, like, you're not like performing surgery. That does not count as husbandry. We're going to talk about what counts as husbandry, but basic care. The other thing that must apply is that student is working with non-living tissue provided by the supervising scientist from their lab or the lab's existing study. This basically means that you're only that you can work with animal tissue provided to you. And honestly, like, most of the vertebrate animals research we're seeing with STS are tissue studies. Sometimes we get these lab animal studies, but because the rules are restrictive, it's a little bit more rare. So this is in here because we want to make sure that if you are working with tissue that's being given to you, that it's coming from a mentor study. And that's because of the next rule, which is that no animals are sacrificed solely or primarily for the purpose of the student's research study. The student, under no circumstances should an animal be sacrificed for your research. You cannot design a study or do something to the animal, and then that is the reason it is sacrificed for then you to look at its brain or whatever it is that you're looking at. You may use tissues that come from a mentor study where you're receiving the byproduct, right? So in that situation, we're going to ask you in task 3 of the application to demonstrate proof of their IACUC permissions and kind of the dates of when all of that took place. The permission was granted, the research study was granted. Both you and your mentor are going to be asked to verify that that animal was not sacrificed, or those animals, for your study. And there's sometimes some gray area here, and you're going to want to be really honest with yourself. And you can certainly always design great studies using the tissues that are given to you. You don't have to design that initial research question that results in the death of the animal. But we don't want high school students out there causing harm to animals in this way. So you cannot design an experiment even with your mentor and then later sacrifice the animal to see the impact. You can only use the surplus materials from the mentor study. And then the last thing that applies, student was not involved in the collection of data directly or indirectly where the research involved invasive or intrusive experimentation that causes more than momentary stress or pain to the vertebrate animal. This one's pretty self-explanatory. There are some other rules associated with it here. What counts as like an invasive or intrusive experiment? Some examples, anything that causes stress or harm. Or could cause stress or harm, or could lead to death, right? So, oh, I lost my slide. Some examples are if you separate mothers and babies, that's going to cause a lot of stress for both parties. That kind of study is not allowed. Toxicity studies with the intent to study toxic effects of a substance on a vertebrate animal, like you're intentionally causing harm, or you could cause unknown harm, so we don't allow that. Anything else, like a predator or prey experiment where you're like literally the animal like could get eaten or could be very stressed at that point, that is also disallowed. So those are some examples of studies that students could not design with live animals. So again, to reiterate, the student can only work with live vertebrates at an RRI, never a home, never a high school, and they can only really design very simple studies that require RRI supervision and IACUC pre-approvals. You cannot design studies that require animal sacrifice or inflict harm. And again, we have to have the RRI requirement because unlike ISEF, we do not have a mechanism like a local science fair that can pre-approve your project to make sure that what you're doing is safe before you do it. So the RRI serves that purpose, which is why we require that you work at one. It's just because the programs function differently, and it's also the reason why for STS, as many students from a certain high school or region can apply. We have no restriction on that because we don't have that initial filter of a local science fair process. So it's kind of, give and take. You can't have your cake and eat it too, and that applies in this case. And so the RRI is going to oversee what you're doing if you get approval from them. Some things that you could do, if you're wanting to work with live animals, it doesn't mean you can't work with them at all under the correct circumstances. There are some low risk types of projects that tend to get approved very easily, that's student designed to work with a live animal under the supervision of the RRI. Sometimes like a maze project, and that's we've seen studies where students help with animals under veterinary care, and they're collecting data and doing all kinds of interesting things. So no, there are projects that you can do so long as they meet these requirements.
Another question that we see that can sometimes trip our student entrance up is really what is husbandry and what really falls under that definition. So basic husbandry is defined as the regular care of research animals. So that might look like providing food and water to an animal, cleaning enclosures, monitoring their health, maintaining their equipment, and other daily care tasks. What that does not include is the administering of experimental procedures, performing invasive surgery, or again, as we've mentioned before, introducing new stimuli to the environment. None of those are covered under basic husbandry.
Like we said before, to do a project that the student has designed for vertebrate animals live at an RRI, you will likely need to obtain IACUC pre-approvals. You will need a mentor, and you will need to go through the RRI's existing IACUC process. We don't have a mechanism like we do for IRBs. If you're doing human research, you can make your own IRB at the high school. That's not a thing for STS. You're going to need to get proper IACUC permission from the institute at which you are doing this work. Now, you might not need to go through yourself if your project falls under your mentor's pre-existing IACUC. So this very well could be the case if they are giving you tissue that they took from an animal post-death, or if they're giving you data from a study that they're doing on live animals. So talk to your mentor. Depends on the circumstance what you might need to submit to us. And if you did anything that falls in this category, you are going to be asked to show proof of like pre-approvals or your mentor's pre-approval for these studies within our application.
So we did also mention that there are some limited cases where you can conduct a project in a natural environment involving live animals. So these kinds of studies can be conducted in the field or at another site as long as there is documentation of IACUC approval, wildlife permits, other proper permissions and licenses, and the project you're doing adheres to all of the following. So again, that is every single one of the ones that we're going to read here. So one, this study is observational or behavioral. So again, you're just observing, you're watching animals' behavior. The study is non-invasive and non-intrusive. It does not affect an animal's health or well-being by causing distress, discomfort, or pain. And the student has no direct contact with the animal unless performing basic husbandry or weight measurements without introducing new stimuli to the environment. So any of these projects that only involve the observation of animals in their natural environments, where there are no experimental or behavioral procedures being conducted, and where the study isn't really affecting the animal's health or their well-being by causing any kind of stress, discomfort, these aren't going to require prior review and approval. Again, that's as long as you have not manipulated the environment in any way.
That's right. And I do want to add something here that we referenced at the top of the session, which is that there's an argument sometimes that the school or home counts as the natural environment. Sure, that could be the case if you wanted to do something where you're observing your pet dog. We do see projects where students are interested in working with their pets. The big caveat here is like you can't do anything with them. If you count, your house is the natural environment for your pet, yeah, sure, I buy it. However, like you can't change their food, you can't put a cast on them and see how they walk, you can't test your device on them, can't do anything. So that does limit the opportunity for your study. Same with fish. We get this one a lot. Fish in the home environment is the tank in your house. Great. So you're counting that as the natural environment. Can't do anything to it, right? Like that is their home. You can't put a different food in there and see how they react. You can't change the pH. Can't do that. So it again limits like what are you going to study and do? Unless you're looking at a variable like time, which changes throughout the day, and you're looking at their behavior. I don't know. I'm sure you could still think of a cool experiment, but know that you're limited because like the whole point of that counting is that you can't do anything to them. And we get a lot, something we've disqualified quite a bit in recent years are vertebrate research projects from the high school in a high school setting. So know that the high school lab unfortunately does not count as an RRI for STS. Okay, so you want to work with vertebrate animal tissue. What do you got to do? Reminder from the top that any tissue that you're using must be from another study. It cannot be from a study that you designed while the animal was living. Pretty much if you're using like organoids or something like that, it can't come from an animal that has been sacrificed because of you. That's called design to kill and you will be disqualified. If you are using the tissue that's left over from a mentor study, like we said before, that's fine. You'll just have to show the proper documentation. The projects that do use vertebrate animal cell lines and tissue will need to show the source of their the source. And the source could be many different things. So it could be the circumstance that we've talked about several times now, where it's coming from a mentor study. But there are other places you could obtain tissue and cell lines that you're using for your study. And one of those would be a commercially available catalog like ATCC, where you can purchase or your mentor can purchase a commercially available cell line. That kind of study does not require any pre-approval. You will have to show us documentation of where you obtained that cell line, but that should be pretty easy to do. It's a purchased line. You can also obtain data, cell lines or tissue from like a bank, like a blood bank, that kind of thing. And again, that's a publicly available source. I mean, you have to have the right permissions and obtain it, but it's meant for that purpose. And so you will have to demonstrate, again, like some kind of receipt or email proving where you got it. And then we also have some rules around projects using data and images pertaining to vertebrate animal studies. So this again comes down to the source. Were the data or images of the vertebrate animals coming from a publicly available study or database? Great, does not need pre-approval. You can just show us documentation about where you got it. Open source, public, anyone can use it. If you are using data or images from a study that is not published, like your mentor study, you will need to show proof of their IACUC and of and documentation as well that they or you can show like a publication because if they've had the research published. The other thing you might need to demonstrate is any proper licenses to work with certain types of animals. And that's pretty much it. So you can always go through task 3 of our application if you have questions about this, but the big picture down, if you're working with vertebrate tissue, it matters where you got it. Did you get it from a mentor? Did you get it from a published source? And then that determines like, did you need pre-approval and what kind of documentation you have to share with us.
And so we'll just pause to do a quick note about zebrafish as well, since that's a pretty common organism that we see among our entrants. Zebrafish embryos must be euthanized by the 7-day post-fertilization window. You are going to be asked to verify this information in the application.
That's right. As a research organism, they are not considered vertebrate animals until that point. So it's very important that we are verifying when the animal was disposed of. And yeah, that's a new regulation that we're asking more specific questions about the dates for that. So if you've been around a while, know that that is new. The rule's not new, just the questions around it are. All right, so if all of this sounds confusing, again, you're not alone. It's a lot to remember and a lot to know. We recommend that you check out the rules wizard in task 3 of the online application. Anyone can open an application to go through this exercise if you're considering applying in a future year. We said our rules do change, but there's small changes here and there that enough that this should still guide you towards basically what you'll need for next year. So you are encouraged to do that so you know what paperwork you might need to submit.
And so just to recap and talk about some requirements you'll have regardless of where or what kind of animal study you are doing. One, we will reiterate probably the most important takeaway is that you are just limited in what you can do to live animals here if you're planning to apply to STS. We want to be sure again that you are safe and that the animals are safe. And so just be very sure to read the requirements and make sure that you are adhering not just to one or a couple of them, but to all of the requirements to make sure that your study will not fail to qualify. You also, another good thing to reiterate, you can't design a study that will require an animal to die. So there is no designing to kill. The project that you submit to STS must start with non-living material. Another thing we'll always ask you to do is to demonstrate documentation of where your tissue comes from. So make sure that wherever you are being asked within the application, especially in task 3, we want to see documentation of any pre-approvals, of links to databases that you used, receipts, mentor letters, whatever is required for your specific study. Make sure that it is all in there and all properly signed and dated. Last thing here we'll mention is make sure that if you are working with cell lines, you are also following all of our PHBA rules, and you'll likely need to complete a risk assessment as well.
That's right. Now we have a note for any mentors who are watching today. Some tips that we have for you as you support students is to, one, save any documentation or paperwork related to the work you're doing with the student. This is a huge pitfall that we see in the application process is a student might, wait till the very last minute to request their recommendation. Hopefully they've done that early for you, so you have a bit of time. But then sometimes there's a scramble to find, oh no, we need to pull a copy of the IACUC or whatever it is. So if you have that kind of ready to roll, then you'll be ready whenever the student does ask you. This information is important and it can save the student if we're like worried that they might not have the proper permissions or they, it's questionable with what the answers that they have provided to us. We tend to trust the mentor. So know that you can help them out a lot by doing that. We do recommend that you start your we suggest rather that you start your recommendation early. As soon as your student opens an application, tell them like, hey, submit that form for me. I got to get in there. Sometimes they're hesitant to submit the request early because they're like, I'm still working on my project. And but for the most part, you're going to be able to answer 90% of the questions in the recommendation form. Note that it is not a letter of recommendation, it is a form with very specific questions about what they did with you, some questions pertaining to our rules, that sort of thing. So it might take a little bit longer than you think. You can help the student with the rules wizard, task 3. Again, this is where we're collecting a lot of paperwork from them. So they might, it'd be great to have your support with that, or to at least be prepared to answer questions that they have for you about it. And then again, read the rules annually. They do change, especially if you have students enter every year, you'll want to know what's new. All right, now we have a couple of common scenarios that we'll run through as quickly as we can, maybe back and forth, and see how we do. Of the kind of some common questions that we see where students aren't quite sure what to do or how the rules might apply to them. So I think we'll just buzz through. I can take the first one, which is I collected feathers for my project. What permissions do I need? This really none. You're working with byproduct of things you found in the wild. That's kind of found tissue. You could call it. It's not commercially available, but it's not something you stole or took from the animal. You sometimes if you're taking things from the wild, these will count as PHBA studies because you're studying something unknown, but it kind of depends what you're doing with the feathers. Are you measuring them? Are you examining them for color? Are you I don't know what you're doing with them, but you don't need any special permissions.
All right, next one. I want to give my dog some supplements. This would not be permitted under STS rules. This is one of the examples where you are changing something in the environment. So you're giving your dog a new supplement. For us, that is a project that you're not able to conduct in the home environment. Anything you do with a live animal is really going to need to be, again, conducted in an RRI under a lot more supervision than just your home environment. So not for STS.
That's right. And it's very unlikely that if you did this study at an RRI on, I mean, I don't know if your dog now lives at the RRI or other dogs that are there, it may not get approved by NIACA because that technically counts as a toxicity study and of unknown effect on an animal. So probably unlikely. The next one, I am using tissue my mentor gave me from a study we conducted together. This one's got a lot of gray area, so we're going to probably want you to give as much information in the application as possible about who designed the study. Certainly you can design parts of the study together. If you have sacrificed an animal to obtain this tissue, then yeah, we're going to have some questions about that. So it's very possible that, if the mentor designed the study and this is like that their lab does, that it's going to be okay. But if like you and the mentor are like, oh, the student, this was really their part of the project. They had the idea to like, give the animal this food or whatever it is, and then see what happened. And then we sacrificed the animal. We like looked at different organoids. Then that would not be allowed because the student had the idea. And then the animal was sacrificed for what the student designed and did to them for their study, and that's not allowed.
I am using fossilized bones. So this is a little bit like, I think, our feathers example before, where it's not something that you are taking from an animal. I think we would definitely want to see some documentation of where you have obtained these fossilized bones, at what study you are using them for. But I do think this would be permitted under STS rules.
Yeah, totally. If we see some projects each year where students have like gone to a local museum and are like doing something with mammoth bones or whatever, that's cool. And what we require is just documentation of like you had permission to do this and that they were fossilized and they came from this I guess counted as a commercially available source, even though you need special permission to get in there. I am using a commercially available animal cell line from ATCC. Great. This is pre-approved. You don't need, or this is exempt. You don't need any pre-approvals for this kind of a study. You do need to show us documentation of where that cell line came from. So just the receipt, if you have it, sometimes the student will obtain this commercially available cell line. The student won't buy it, but they'll obtain it from their mentor who's had it for a few years. So sometimes we'll accept a letter from the mentor about their origin of this cell line, but a receipt is always preferred.
I am studying pregnant cows. So this is one where you would need to be conducting this research at an RRI. And again, your physical contact with the pregnant cows would need to be limited to basic husbandry. But we know this can trip some folks up. What is basic husbandry when there is a pregnant animal involved? So in this case, things like ultrasounds, those would actually be permitted. Anything that is normal in that research environment for that research animal is permissible by STS rules. But again, we are going to want to see some documentation there, just of your pre-approval and your role in this study.
And a lot of RRIs will have limitations to what high school students can do anyway. So that sometimes naturally limits you to like what is allowed. I observed birds and changed the food around them, see how they would behave. Anytime you're doing a study in the wild, you can only observe. If you were doing anything to stimulate their environment that is not allowed, this project would be disqualified.
I observed birds and did not do anything to their setting, just observe them at different times of the year. This is totally fine. So in contrast to the previous example, nothing is being changed here. The only thing that is changing is the time at which you are conducting those observations. So this would be totally permissible.
I weighed fish and frogs in nets. This is an interesting one, and it really depends on making sure you have the right permissions and context. So most studies done in a natural environment need to be observational. And this is too, because in this, you're technically observing their weight. But we do have students who are doing citizen science projects, and they're helping to collect the data that they're using. And you're collecting fish in a net for that and then releasing. If that is all you're doing, that's okay. However, you need to have a wildlife permit or show a permit from the the larger entity and project that you have permission to do this. So those are the circumstances where that kind of a study is allowed, but you can only be really doing that.
I can only give you an IACUC renewal paperwork, so as opposed to the original studies paperwork. This is fine. I think we'll want to see dates of the original study, but if all you have is the renewal paperwork, that is a-okay.
Yeah, the original is always preferred. And if you have renewal and original, even better, because sometimes that demonstrates that like, if there's a continuity concern of like, you kind of started in the middle of like when the first one expired and then when the next one started, we want to make sure everything's on the up and up. But like Elaine said, if the renewal has enough information, then that's likely going to be okay. But you want to make sure it has enough information. Like if it doesn't include like your mentor's name or like what you're doing or what animal you worked with or that your mentor worked with, that might not be enough information for us to go on. The next one is I performed surgery on the animal. That's not allowed under any circumstance. Even if you're at an RRI, the student should not be performing veterinary care besides anything that counts as husbandry, and surgery would not count as husbandry.
Next one is I sacrificed the animal. Likewise, not permitted under STS rules. No euthanasia of your research subjects is permitted.
I studied fish in a tank at home or school. If you are only observing the fish in the tank in your home at school, that is fine. But if you are doing anything else, changing the environment, changing the food, then that is not allowed per STS rules. That needs to happen at an RRI.
Gave animals different medicines, but an IACUC did review my study.
We would want to take a look at that. So if you're in that kind of situation, you do discourage those kind of studies for sure. But if you are, working with a mentor and that is what they do, and you're working with data that comes from it, then that situation might be allowed. But again, comes down to like some of these circumstances are very specific, and our team is always happy to take a deeper look with you into your project or your plan to say like that's against the rules or that would be fine.
All right, so we know this was so much information. As always, we do have so many other resources in addition to this webinar available to help you. One great thing to take a look at is our common FTQ document. So that is reasons students have failed to qualify in the past. Just good to skim and review, make sure that you're not falling into any of those really common pitfalls. Another way to avoid those is obviously, of course, making sure you have read our whole rules book top to bottom. That has everything you need in order to make sure that you are getting the right permissions and pre-approvals if necessary to do your project. And if you have any questions about that paperwork, you can visit our application at sciencetalentssearch.smapply.org. Pop into task 3, that is our rules wizard. It will walk you through exactly what you need in order to know what kind of documentation we're going to want to see for your particular project. If you have any questions that we didn't answer today or that you're not comfortable for whatever reason popping into our comments, you're always welcome to contact us directly at sts@societyforscience.org. A member of our team will get back to you with a response within usually about 3 business days this time of year. But always happy to help, and definitely do feel free though to pop your scenarios, questions in our comments. We'll be back next week with our final rules mini session, and we'll make sure to answer those questions then too.
That's right. Thank you all so much for joining. Have a great day. Good luck.